Thank you.
I can, this is the part of the centenary celebration. So I joined the UC College in 62. That means the college was only 41 years old at that time.
Hello, Sarah.
Hello.
Karen, എങ്ങനെയാണ്?
Go on, sir.
Yeah, so I'm, I never thought, I would be able to participate in the centenary celebration. I'm so happy to be part of it.
We are glad that you would come and join us, and you can take the initiative in doing that.
Okay, sure.
Another 2 minutes for our program session. Let's begin our session.
delegates and participants to this August gathering. Thank you all for taking your time out and being here. We are fortunate enough today to have people all around the world, both from the different states of India and all other countries like Canada, Australia, Iraq, and all. And also, we are very fortunate to have different mentors and well experienced persons with us today. Thank you all for being here. And just a reminder, the participants, please follow the instructions during the webinar. Be sure to mute your audio so that there is no disturbance during the entire session. Just a little housekeeping before we get started. If you have any questions during the presentation, please type them into your questions, into the questions box in your Zoom panel or the YouTube panel. We will bring them up during the presentation, or also we will also discuss them after the session. And the feedback form of the webinar only available at the end of the session. Today, we are presenting Zebrafish Disease Models for Phenotypic drug screens and personalized medicine, which is presented by Dr. Anju M. Philip, St. Michael's Hospital, and the Zebrafish Center for Advanced Drug Discovery, Toronto, Canada. And also, we have Professor K. I. Varghese, University of Arkansas for Medical Sciences, AIUSA, and is one of the board of directors, UC College Alumni Association of North America. And we have Dr. Reverend Father Thomas John, manager of Union Christian College, Aluva, and all the dear faculty members and all other active participants. The Department of Biosciences, Union Christian College, welcomes you all to the webinar, Zebrafish Disease Models for Phenotypic Drug Screens and Personalized Medicine, which is conducted by the Department of Biosciences, UC College, Aluva, in association with UC College Alumni Association of North America. I welcome you all. Zebrafish, which are tropical fish native to India, have unique characteristics which they make them an ideal candidate for drug development platforms. They share 70% of our genetic code and have a counterpart for 84% of all genes known to be associated with human disease. Though target-based chemical screens are a focus of modern drug discovery, the effectiveness of this reductionist approach is being questioned in view of declines in the number of drugs that actually reach the market through this approach, especially in the context of complex human diseases like sepsis. Zebrafish therefore have emerged as a leading model for in vivo phenotypic screens for various diseases like stroke, schizophrenia, and sepsis. Zebrafish are also being widely used in conjunction with whole exome sequencing in human to not only confirm gene mutations that might result in a particular illness, but also to immediately test and confirm possible treatment options. So without any further delays, we may step into the official inaugural session. Seeking the blessings of God Almighty, let's begin our today's session.
You are my strength when I am weak. You are the treasure that I seek. You are my all in all. Seeking you as a precious jewel. Lord, to give up, I'd be a fool. You are my all in all. Taking my sin, my cross, my shame, rising again, I bless your name. You are my all in all. And when I fall down, you pick me up. When I am dry, you fill my cup. You are my all in all.
Now, I invite Mr. Shyam Mohan sir, Head of the Department of Biosciences, UC College, for the welcome address. Sir, please.
Thank you, Nidhina. Today is indeed a very special day, 24th October 2020. We, as a part of Biosciences Department, are celebrating our centenary celebrations in the form of a seminar. The most important aspect of this seminar is that we have our former student, uh, Dr. Anju Philip, to deliver the lecture on zebrafish disease models for phenotypic drug discovery. And my job here is to welcome everybody to the gathering. So, first of all, good evening to all the members who are participating in the seminar from India, and good morning to all tuning in from the American continent, Canada, and other time frames. So first, I would like to welcome Reverend Thomas John, the manager of the college, to this gathering to witness the seminar on zebrafish models. Thomas John Achin has been instrumental and guiding us in celebrating the centenary celebration the academic way. So he has motivated us to conduct. We have already conducted one webinar earlier. So I welcome Sir for guiding us and promoting us to deliver programs of this magnitude. Welcome, Achin, to this meeting. Now I would like to welcome Dr. Ike Verghese. Sir was here last year. Sir had come to our college and had delivered a talk on atherosclerosis and drug designing associated with it. And he has been a sort of a father figure for us from last year onwards, ever since we met him. And his cordial relation with us and with Susan ma'am was the guiding light for organizing an event in this time of the pandemic, so that we can look forward to more kind of associations with Dr. Verghese and the UCCAANA, so that we'll have the best speakers for our students and for our college as well. I welcome Dr. Kotail Ike Verghese sir to this event.
Thank you.
Next, I would like to welcome Dr. Susan Epen. Susan Epen ma'am has been a sort of a revelation for us, because she has introduced us to some of the best speakers in India on varied topics, and she continues to press us for delivering the best. And she has definitely been a real role model in guiding us and telling us that we should be at the forefront. The UC College should be at the forefront of conducting seminars of various kind. And in her presence, we have, I think, around in the last year or two, we have had many such occasions where from India and abroad, we have had the opportunity to meet, to talk, and listen to some of the best academicians and scientists. And Susan ma'am, you continue to be our inspiration and keep guiding us. Welcome, Susan ma'am, to this event. Next, I would like to welcome our favorite teachers, and they have been the inspiration. It's very special that you're here, especially Jacob Gurian sir, Thomas Phillips sir, some of our mentors, some of our role models, and the pioneers of the department. It is like under their guidance that we were able to understand what academics, administration, etc. are. And you continue to be the source of inspiration with your wonderful teaching methodologies that you still, and especially Jacob Kurian sir with his reverberating energy, you continue to motivate us big time, sir. So I welcome, I also know that Shirley ma'am is here, and some of all the teachers who are a part of the BTNSP community in the beginning, and later the bioscience department as it is. They continue to come here, inspire us. So welcome, sir. Welcome all of you. Welcome all of the guiding light mentors of our department. We are proud to have you here, and we are proud to see Anju here perform well here. Now I would like to welcome Dr. Anju Philip. She was a student. She was a successful student. She had acquired university ranks. She is one of the best role models our students can have, because she has completed BSc BTNSP here and MSc Biotechnology. She was a university rank holder, and she has excellent publications, and her profile will be viewed to us in detail. And she will, we have already seen the trial version of her presentation, and I'm sure that Anju will give us a very good time, and it'll be a very informative and a very excellent session. And thank you, Anju, for finding out time for your department, and welcome you to this program. Thank you.
Thank you very much.
Next, I would like to welcome the faculty of our department, Dr. Serin Sarah John. Serin ma'am has been coordinating the efforts in tune with Susan Miss and Ike Burgess sir. We have had mock sessions, and she has been the soul of this program, coordinating the students, conducting the associated backstage online trials, etc. Saren ma'am, I welcome you to this program. I also would like to now welcome other faculty from our department, other faculty from our college, and the teaching and non-teaching faculties from our college as well to this program. As a part of respect and as a part of friendliness, I would like to welcome all of those teachers who are involved from our department as well, and who has been helping us in organizing programs throughout these pandemic times. So welcome to all the teachers and non-teaching staff of our department and our college. Finally, I would like to welcome all the participants of this event. That includes students from the Biosciences department, students from other colleges, research scholars, Ph.D. holders, and scientists who are viewing this program. I welcome all of you. Welcome one and all. Thank you.
Thank you, sir. Thank you, sir, for the great welcome. Now I invite Reverend Father Thomas John, manager of Union Christian College, to offer us felicitation and to bless us.
Dear friends, it is a great privilege to listen to Dr. Anju M. Philip, one of our own alumni, especially one who grew up as a member of the UCC family, daughter of one of the architect and builder of the biosciences in UCC, Dr. Thomas Philip. Anju comes to us as a distinguished scientist and scholar on her own right. Moreover, it seems to be a collaborative venture in which some of our distinguished alumni who has been in this field is joining and facilitating from USA. Particularly, I must mention the name of Professor Kotail I. Varghese, University of Arkansas Medical Sciences, USA, and one of the members of the board of directors of the Alumni Association of United States and Canada. It is this sort of facilitation that we expect from our alumni, and we greatly appreciate the same. I know that our adjunct professor, Dr. Susan Ipin, is also there behind the scene, encouraging staff and students and giving leadership for their incubation and startup initiatives. I also would like to acknowledge my appreciation to the HOD, Professor Shyam, and his colleagues in making big strides in the development of the biosciences department. I don't know much about the subject on which Dr. Anju Philip will be talking to you, to us, but she is talking to us about an extremely useful model of human disease that will help us in drug discovery and personalized medicine. As a psychologist, I am very much interested in personalized medicine, as many psychotropic drugs don't have the same effect when given to different individuals with the same classification, like schizophrenia and so on. I do not want to say anything more which will expose my ignorance in this subject. One of the gifts of being a manager is to listen to people from diverse disciplines and learn from them. I hope that Anju's lecture will inspire many of you to take on your studies to greater heights. With these few words, I inaugurate this webinar and wish you an extremely inspiring and motivating time.
Thank you, Father. On this webinar, we are expected to have our principal, Dr. David Sarge Matthew, with us, but because of his inconvenience, he couldn't be with us. So in the absence of his presence also, I would like to welcome him into the webinar. Now, with all the respect, I I invite Professor K. I. Bhuvanagis from University of Arkansas for Medical Sciences AI USA. He is one of the board of directors, UC College Alumni Association of North America, to kindly introduce our chief guest into the webinar. So please.
First of all, I would like to say that I am excited to be a part of this webinar series. I had the privilege, as I mentioned already, I had the privilege of visiting UC College and giving a talk almost exactly a year ago. In fact, it was on October 25th, 2019. I enjoyed my visit, enjoyed the visit very much. I'm very thankful to Dr. Reverend John Thomas, Dr. Susan Epen, Dr. David Matthew, Dr. Shirley Thomas, Dr. Shyam Mohan, and many other faculty for making my visit a memorable one. I also had the opportunity to talk to a number of students who were very enthusiastic about biomedical research. I left the campus with a great desire to continue my interactions with UC College. As this seminar series is organized by UC College in association with UCCAANA, let me say a few words about UCCAANA. An association of former UC College students in North America, that is USA and Canada. It is a tax-exempted, non-profit charitable organization. It was established in 2006, and it was officially inaugurated by Dr. A.M. Chakosar in 2007 at a meeting in New Jersey. Since that time, UCCAANA has been involved in many activities at UC College. Let me mention a few. In June 2011, UCCAANA established an educational endowment at UCC to award scholarships to students based on merit and need. UCCAANA was also involved in establishing a Center for Study in Religion and Society, and also T.O. Abdullah Endowment Lecture Series. Additionally, UCCAANA members established Vidwan P.G. Nair Endowment and Research Center at the Malayalam Department. I do not know how many UC College students are there in North America. The number must be in the thousands, but UCCAANA membership is only over 300. The UCCAANA board is eager to recruit more members and strengthen our interactions with UCC. Therefore, I have a request. If you know any former UC College students, your friends or your family members in North America, please let us know. We will contact them and try to bring them into the association. Now, without taking much time, let me say a few words about Dr. Anju Mary Philip. I cannot think of a better person to start this series on drug design than Anju. She grew up in the campus as the daughter of Dr. T. Thomas Philip, former principal of UC College and the chair of the zoology department. She did her BSc and MSc at UC College in biotechnology. She got the university first ranks at the Mahatma Gandhi University for both BSc and MSc. A highly commendable achievement. After being a guest lecturer at UC College, she decided to go to Canada to pursue her higher studies. And she got her Ph.D. in biology with focus on immunology, endocrinology, and animal physiology from the University of Waterloo, Ontario, Canada. She is currently involved in the drug discovery project at the Zebrafish for Advanced Drug Discovery at the St. Michael's Hospital, Toronto, Canada. She already has 2 drug patent applications on sepsis. Without taking more time, let me ask Anju to tell us about her exciting work. The title of the talk is Zebrafish Disease Models for Phenotypic Drug Screens and Personalized Medicine. Anju, take it away.
Thank you. Can everyone hear me?
Yes, yes.
Yes, yes.
Okay. So I'm going to share my screen. Can everyone see the screen?
Yes.
Yes,
Thank
yes.
you very much. Okay. So good evening to all of you attending this webinar from India, and good morning to all of you attending this meeting from the US and Canada. Firstly, I would like to thank Dr. Ait Wargi's uncle for the very kind introduction. As the Secretary of UCCAANA, I've had the honor of working alongside Uncle for many UCCAANA activities. The President of UCCAANA, Dr. Thomas Matthew Uncle, is also attending today's webinar. I would also like to thank the manager of our college, Reverend Thomas Jonathan, and head of the Department of Biosciences, Mr. Shyam Mohan, for their very warm welcome and kind words. I would also like to take this opportunity to congratulate and thank the organizers from the Department of Biosciences, Susan Epen Madam, Serene Madam, and Shyam Sir, for organizing this webinar series in connection with the centenary celebrations of our college. So I'm really, really excited to be here today, and I consider it a privilege and an honor to be the resource person for today's webinar. Without further ado, I will move on to my presentation. Zebrafish Disease Models for Phenotype-Based Drug Screens and Personalized Medicine. And these are all images of zebrafish embryos that you see on this title slide. They're all still within their chorions. So this is an overview of my presentation. Firstly, I'll talk a little bit about myself, my background, my interest, what I do today, and share some memories about my time at UC College. Then I'm going to talk about zebrafish phenotype-based drug screens. And here I'm going to talk a little bit about zebrafish in general, and then talk about the differences between phenotype versus target-based screens. Then I'll talk about some zebrafish disease models that I have worked with. And finally, I'll tell you a story from start to finish, my zebrafish sepsis model story. The third part of this talk will look at zebrafish, the use of zebrafish in I'm going to share with you 2 collaborative research projects that we did in our institute, broadly looking at sialic acid metabolism and genetic diseases. So about me, this is a photograph taken in 1988 at my father's doctoral convocation from the University of Guelph in Canada. So I was about 3 years at the time. I studied in Canada till grade 3, and then we moved back to India. So I did most of my schooling in Kerala. From 2003 to 2006, I was a student in the BTNSP program here at UC College. And from 2006 to 2008, I was a student in the MSc Biotechnology program. So this image here shows my MSc project that I did here at UC College under the supervision of Praveen sir, who many of you might be familiar with. So I used a technique called PCR RFLP analysis of mitochondrial DNA to trace the origin of meat and meat products. So this technique is unique in the sense that because we are isolating mitochondrial DNA, you can even trace the origin of even highly processed meat products like meat cutlets and meat rolls. So you isolate mitochondrial DNA from pure meat samples like chicken, beef, pig, goat, etc. And also from highly processed meat samples like meat cutlets and meat rolls. And then you amplify a portion of it with PCR. And then the amplified product, you will subject it to restriction fragment length polymorphism. You'll subject it to restriction enzyme digestion. And then you'll run the products on a gel. So from this gel picture, you can see that the banding pattern seen in meat cutlets is identical to that seen in bovine meat, proving that these cutlets are indeed of bovine origin. So this technique finds widespread application in detecting food adulteration. Then from February 2009 to August 2009, I was a part-time guest lecturer here at UC College. And it was during this time that I developed a more personal relationship with my teachers in the department, especially with Jyoti Lakshmi Miss. Then in 2009, I moved to Canada to pursue my doctoral degree from the University of Waterloo. And this is a photo taken at my convocation from Waterloo in 2014. So you can see my husband, my older son, Alex, and my parents in this picture. So for my PhD, I worked on studying the crosstalk between stress-immune growth processes in fish. I identified that stress can upregulate a class of genes called the SOX genes, or suppressors of cytokine signaling, and the SOX genes in turn can negatively regulate immune signaling as well as growth hormone signaling. So SOX genes are a connecting link between stress, immune, and growth processes in fish. Now, I'm going to talk about what I do today. So I would call myself a researcher, an entrepreneur, and a full-time mom. So since 2014, I've been working at the Zebrafish Center for Advanced Drug Discovery. And the stuff I'm going to show you today is from my work there. From 2016 onwards, I am also the managing director of an animal hospital here in Canada. So my husband is a veterinarian, and we started this animal hospital in 2016. This is a news article that came in Facebook that ranks our hospital one among the top 3 in the area. And from 2011 onwards, I have been a mom to 2 wonderful boys, Alex and Philip, who constantly manage to keep me on my toes 24/7. Next, I'm going to share with you some UCC memories. So my association with Union Christian College, SUTEC Association, started way before I even became a student of the college. And this is a photograph taken from one of our one-day trips to Tumburmuri and Ilithod. So as kids of the professors in the SUTEC department, we used to tag along on all these one-day trips. And on the right, you can see all of us, the kid gang that accompanied this trip. In fact, our Shyam sir was actually a BTNSP student at this time. And here I've shown you some pictures that show you some activities that I was involved in while I was a student at UC College. So the first picture here to the left is from a public awareness seminar that I gave in connection with World AIDS Day. You can see the HIV model that I made on the table here. And in the front row, you can see Jacob Kudian sir and Annie George teacher. So Jacob Kudian sir was very, very instrumental in the development of our department. And I would like to remember that at this time. Then I was also quite involved, active in SCM activities while at UC College. And this picture here was taken at an SCM retreat at Ailey Hills. And the picture shown on the bottom is from a SUTEC Day Valentine's Day skit. So I used to write a lot of skits when I was in UC College. And in this skit, I'm also acting in this skit. So I'm the boy in the blue hat in this particular skit. So my take-home message from this slide is that you can be and become whoever you want to be, as long as you have the determination and the heart to do it. You can do research, you can start your own business, you can have a successful career, you can be a wife, and you can be a mom all at the same time. You don't have to give up on any one of these things. You can have it all. But of course, it's going to be a little bit challenging because you're balancing 4 or 5 different roles at the same time. But trust me, when you look back, you're going to feel good about yourself because you know that you are trying to make full use of your full potential. And to me, it is definitely worth it. Now we're going to take, and to conclude this section, I'm just going to show you 2 pictures, one from our BSc BTNSP Hyderabad trip, and one from our MSc Biotechnology Delhi trip. Now, so this concludes the first part of this presentation. I hope I haven't tired you out already, because we're just going to start into the zebrafish part right now. So zebrafish phenotype-based drug screens. So zebrafish or Daniel Vario are freshwater fish. They're native to the Himalayan region, so they're Indian. It is a popular aquarium fish, and it is frequently sold under the trade name Zebra Daniel. The top photo shows a male and female zebrafish. So male fish are longer and more darker in color, while female fish are more chubbier and they have a bigger underbelly because they carry a lot of eggs in their belly. Below that, you'll see a picture of zebrafish embryos, and you can see that they're all within their chorions at this stage. On the top right, you see a transparent zebrafish. It's called a Casper line. And it's transparent because out of the 3 skin pigments that you normally see in zebrafish, this one only has 1. So that is why it appears transparent. And underneath, I'm going to show you a video of zebrafish development. So going all the way from 1-cell stage to 3 days post-fertilization, when they will hatch out of their eggs. So I'm going to play the video. So you can see the cells dividing. Then you can see the different stages of embryogenesis: blastula, gastrulation, the head forms here, the tail at this end, and you can also see the muscle somites developing. And now you can see the zebrafish moving around a lot inside the chorion. That's what, because it's trying to hatch. And at 3 days post-fertilization, it will finally hatch out of its chorion. Now this figure shows representative images of the different stages in zebrafish development. So the 1-cell stage, then the different stages of embryo development, 24 hours post-fertilization, you can see that it is still curved because it's still inside the chorion. 72 hours is when it hatches out of its chorion. And here you can see an adult zebrafish. An adult zebrafish is about 2.5 inches long, while zebrafish embryo is about, or larval stage is about 3.7 millimeters long. So it's just a few millimeters. Embryogenesis is completed in 50 hours post-fertilization, and the fish reach reproductive age in just 3 to 4 months. So the power of zebrafish lies in the following facts. It is inexpensive and easy to maintain. Each pair of zebrafish, so each male and female, can generate over 100 eggs per week. So imagine that big sample size that you get to work with. Zebrafish eggs are fertilized externally. Zebrafish embryos develop rapidly and are transparent. The newly hatched zebrafish can fit into the wells of your 94 well plates or even your 384 well plates because you're just a few millimeters long. Humans and zebrafish share 70% of the same genes, and 84% of all disease in humans have a zebrafish counterpart. So zebrafish genome has also been fully sequenced, and it is particularly notable for its regenerative abilities, and zebrafish have been modified by researchers to produce many transgenic strains like the ones you'll see in the following slides. So a highlight of zebrafish research is the ability to perform whole body real-time imaging. Real-time imaging plus the use of transgenic lines is just mind-blowing. It's amazing. And I'm going to show you 2 videos to prove my point. So these zebrafish that I've shown here are double transgenics. So it's a FLIC1 GFP GADA DS red line, meaning that the blood vessels are shown in green, are labeled in green, and red blood cells are labeled in red. So this first video that I'm going to play is going to show you the heart pumping blood into the circulation. So zebrafish heart are 2-chambered. So they have an atrium here and a ventricle on top. And you can see the red blood cells moving through the heart and into the blood vessels. On the right, you can see the major blood vessels in zebrafish. So this is the dorsal aorta on top, the posterior cardinal vein here, And all these small blood vessels that you see here are called intersegmental vessels, and they run throughout the body of the zebrafish. Next, we're going to take a look at the common techniques that we can use for gene manipulation in zebrafish. So the two common techniques that we normally use are gene knockdown using morpholinos and gene knockout using CRISPR-Cas9. So morpholinos are synthetic single-stranded nucleic acid analogs with a methylene morpholine ring backbone that you can see here. And this ring replaces the sugar phosphodiester backbone that you normally see in DNA or RNA. So morpholinos can be injected at the 1 to 4 cell stage. Ideally, we do it at the 1 cell stage. And morpholinos can bind specifically to its target mRNA or pre-mRNA. And depending on whether the morpholino blocks that binds to the translation start site or a splice donor or acceptor site, it can cause no protein or it can cause a defective protein to form. But the problem with morpholinos is that it is not a stable genetic mutation. Its effects wear out in 4 to 5 days. So you can use it to study gene function only in the larval stages of zebrafish. If you want a more stable genetic manipulation, you need to do CRISPR-Cas9 mediated knockouts. So these are the two scientists. I'm sure you're familiar with them because Doudna and Charpentier were recently awarded the Nobel Prize in 2020 for their work on CRISPR-Cas9 technology. So essentially, first thing you do is you make a guide RNA, which matches the piece of DNA that you want to modify. And then you inject it into the embryo along with Cas9 endonuclease, which acts as molecular scissors. And it will make double-stranded cuts in your DNA. Once the double-stranded cuts are made, your DNA repair enzymes in the body take over. So you can either get non-homologous repair, leading to the formation of insertions or deletions. Alternatively, if a synthesized DNA template is introduced, you can get targeted genome editing. And like I said, this is a more stable form of genetic manipulation. So you can follow the mutated genes right through the larval stages as well into adulthood in zebrafish. So for both these techniques, you need to do microinjections. You need to inject into the zebrafish embryos. And this is the apparatus that we use to do microinjections. So you can see the needle here. On the left, you can see the mold. It's an agarose mold. And this is where we line up all the zebrafish embryos so that we can start injecting them. And you can see all the embryos lined up in this mold right on the left. And then finally, we inject right into the embryo. So you can inject either into the yolk, that's what's shown in the bottom picture, or you can inject directly into the cell at the 1 cell stage. So for RNA, we prefer to inject into the cell. And for morpholinos, you can do injections into the yolk. Now we're going to talk about some zebrafish disease models that I have worked with. So the first model is a chemically induced zebrafish disease model. And it's a chemically induced ICH model, intracerebral hemorrhage model. And the chemical that I used was atorvastatin. So atorvastatin is a statin, and statins are drugs that we use to lower cholesterol levels. However, there are some side effects associated with its usage, including an increased risk for bleeding. And intracerebral hemorrhage is a devastating condition with limited treatment options. Spontaneous cerebral bleeding is a characteristic of the human condition, and it is very difficult to model this in rodent models like mice. So that is why we decided to see if we can try to model this in the zebrafish. And you can see that using atorvastatin, you can induce spontaneous brain bleeds in the brain of these zebrafish larvae when compared to the controls. And these ICH larvae also show increased brain cell death and a persistent locomotor deficit, which is similar to what we see in ICH patients. The next model that we're going to look at is a knockdown disease model for schizophrenia. It's a microRNA 137 knockdown model for schizophrenia. Schizophrenia is a highly heritable psychiatric disorder that consists of episodes of psychotic symptoms like auditory hallucinations and paranoia. And genome-wide association studies have shown that a variant in the microRNA 137 is strongly associated with schizophrenia. But we couldn't model this in mice models because it's an embryonically lethal mutation, meaning that if you try to knock down or knock out microRNA 137 in mice, all the mice die in utero. So you cannot study this mutation in mice. That is why we thought, okay, let's try and see if we can knock down this gene in zebrafish and study it in zebrafish. We noticed that as soon as we knock down the microRNA 137 in zebrafish, these larvae produced gross abnormalities like in the head as well as in the heart. And again, these two organs are affected in schizophrenic patients as well. When we did immunostaining, so we stained for interneurons in the brain, and you can see that in the controls, there's a nice symmetrical pattern of interneurons. But when you look at the schizophrenic larvae, you don't see that nice symmetrical pattern. And also there's overall less number of interneurons as well. And this is also consistent with postmortem studies of human brain from patients with schizophrenia. The final model that I'm going to show you here is a factor 13 morpholino-induced leakage model. So factor 13 is a plasma protein involved in the blood clotting cascade. And recently it has been shown to play an important role in maintaining blood vessel integrity. And its deficiency has been associated with subarachnoid hemorrhages. So we wanted to see if we could model this in the zebrafish. So we knocked down factor 13 using morpholinos, and to check for blood vessel integrity or stability, we injected colored dextrin, and in this case, it's purple colored dextrin into these fish. And then we looked for indications of vessel instability or vessel leaks. So what I want you to concentrate on is the space in between these blood vessels. So you can see the ISVs or the intersegmental vessels in green. Concentrate on the space between these blood vessels. So you can see that it's dark in both these fish, the control as well as in the morphins. So it's like black in both these fish. Even the space in between the vessels in the brain, it's also black in both these fish. So now I'm going to play the video, and you can see the dextrin circulating through the fish, and watch that space that you see. You can see that in the controls, all the body cavities still look black. All the spaces in between the blood vessels still look black, whereas that space is filling up with dextrin in the morpholino-injected fish. So this shows that the blood vessels are indeed unstable, they're leaky, and that's why the dextrin that you injected is leaking out. So far we talked about various ways in which we can generate zebrafish disease models. It can be chemically induced, or it can be through genetic manipulation, like the use of morpholinos or CRISPR-Cas9 system. So the next step is to know how we will use these models in drug screens. And typically we use zebrafish in phenotype-based drug screens. So I'm going to tell you why we use it in phenotype-based drug screens and the difference between phenotype-based drug screens and target-based drug screens. So the traditional drug discovery approach uses target-based drug screens. You start at the target. So you first identify. So let's the protein that in Parkinson's disease. Then you do target validation and assay development, and typically you will do this in an in vitro cell system. I'm sorry. Typically you will do this in an in vitro cell system. And in our case, in studying Parkinson's, we would typically use neuronal cells. Then you use these cells, because it's cells, you have a rich supply of them. So you can use them for high throughput drug screening. You can screen them against hundreds and thousands of drug compounds to identify hits and leads. The problem with this approach is that nowhere in this pipeline are you using an in vivo system. You're only testing the compounds in vitro systems. We finally decide to test them in vivo system. Many a times you'll find that the drug will not work. And this is because of the complexity of the in vivo system. In an in vivo system, you have so many different biological processes happening. So many of them interact with each other. So in such a case, these drugs might not be effective in an in vivo system. And in very rare cases, the drugs might act a little too well in the in vivo system, meaning it will cause a lot of toxic side effects. So either way, it's not good. And that is why drug discovery researchers are now shifting their focus to phenotype-based drug screens. So in a phenotype-based drug screen, you model the whole disease phenotype in an organism. And usually it's an in vivo model. You can model it in mice, but the problem with mice is that you cannot use it for high throughput drug screening. You cannot screen hundreds and thousands of compounds in mice. It is just not financially feasible. So that is where, so you start at assay development and then you end at target deconvolution or the identification of the target. And that is where Zebrafish is important because it combines the best of both the systems. It is an in vivo model where you can perform high throughput drug screens. So now I'm going to tell you a story from start to finish. And this is my Zebrafish sepsis model story. So I'm going to give you a brief introduction to sepsis first. So sepsis is an uncontrolled, dysregulated immune response, which progresses very rapidly to multi-organ system failure, septic shock, and death. It affects more than 30 million people worldwide every year, and it has a mortality rate of 50 to 80%, depending on which part of the world you're in. So sepsis strikes the young and the old alike, and there have been over 100 failed clinical trials in the past decade or so. Therefore, sepsis research is referred to as the graveyard of drug discovery. And intervention strategies for sepsis still remain largely supportive through the use of antibiotics, fluid therapy, and so on. Sepsis pathophysiology is actually a continuum of different pathophysiological states. A patient is diagnosed with SIRS, or systemic inflammatory response syndrome, when he presents at the hospital with 2 or more of these symptoms. A patient is said to have sepsis when he has SIRS along with a confirmed case of infection. A patient is said to have severe sepsis when he has sepsis along with organ failure. And a patient is said to be in septic shock when he has severe sepsis along with hypotension. So I broadly grouped the hallmarks of human sepsis into different categories. So the major hallmarks associated with sepsis are vascular leakage and edema. This means that your blood vessels become leaky. Immune cell activation and the release of inflammatory mediators like cytokines and reactive oxygen species. The maldistribution of microvascular blood flow, the activation of coagulation cascades, and widespread tissue injury and organ dysfunction. Now, why do we need novel drug discovery strategies for sepsis? So there are several reasons for drug failure in sepsis. Like I said, there have been over 100 failed clinical trials for sepsis. And the main reason for that is the complexity of the disease. More than 3,700 genes are differentially regulated in sepsis. So your target-based screening approach is not going to work for sepsis. And when you take drug discovery in general, 9 out of 10 drugs fail to gain government approval. So if you use the target-based system for drug screening, 9 out of 10 drugs will fail to gain government approval because they lack pharmacokinetic properties, high toxicity, and low efficacy when you actually test them out in vivo models. That is why we thought that an in vivo animal model suited for high throughput drug screening would be the best to use in the case of complex diseases like sepsis. Hence the Zebrafish sepsis model. So my project was divided into 2 parts. The first part was developing the model. The second part was using it in high throughput drug screens. So how did I develop the model? So male and female zebrafish were bred, and like we heard, they lay hundreds and hundreds of eggs at a time. At 3 days post-fertilization, when they hatch out of their chorions, I put them in LPS, or lipopolysaccharide. Now, lipopolysaccharide is a molecule that's seen in the outer membranes of gram-negative bacteria, and I used LPS for 2 reasons. The first reason is that more than half of all sepsis cases are caused by gram-negative bacteria. The second reason is that the more deadly forms of sepsis, or the sepsis that leads to death, is usually caused by, or more cases in this case are caused by gram-negative bacteria. Once I induced sepsis in these larvae, I characterized the hallmarks of human sepsis in them. So I looked at all the hallmarks that I mentioned in my previous slide. I characterized each of those hallmarks in these larvae. And if you want more details of that, you can refer to this paper. The next step was using this sepsis model in high throughput drug screens. So once I induced sepsis in the larvae, I put them in 96 well plates. So 1 larvae per well, and screen them against hundreds and hundreds of drug libraries. So each well will get 1 drug. And the purpose is to identify rescue drugs so that they can be further validated in mice and human models of the disease. Now, for any high throughput drug screening approach, you need to have readouts. So readouts, it's what you're measuring. And so I use 3 readouts: vascular leakage, so measuring how leaky your blood vessels are, ROS production, and mortality. So if any of my molecules, small molecules, can rescue all 3 of these readouts, I would consider it a hit in my screen. So now we'll take a look at those results. So this is the control larvae on top, and this is an LPS-treated larvae. The first thing I noticed was that their tail fin swells up. So you can see this tail fin swelling in the LPS-treated larvae that gets worse over time. So this is 2 hours post-treatment, 6 hours post-treatment, and 9 hours post-treatment. To visualize this even further, I used a transgenic zebrafish line in which the blood vessels are labeled in red. And then I injected them with green phytidextrin. So phytidextrin is just 4 kilodaltons. And this, you can see in the LPS treated group, the green phytidextrin leaking out of the red blood vessels. And it's this leaked out phytidextrin that moves all the way to the tail fin and accumulates there, causing that swelling in the tail fin area. The next readout was ROS production. So you can see that the LPS treated larvae, the whole body has systemic ROS production, and it lights up in green when you add a ROS indicator compared to the controls. The third readout was survival. And again, you can see that the LPS treated larvae, if you do not provide any intervention, they will all die off over time. So by 24 hours post-treatment, pretty much they're all dead. And again, this is something that we really want to model because sepsis causes such a high mortality rate in humans as well, 50 to 80%. So next, I'm going to show you the results from the high throughput drug screening. So like I said, these 3 readouts are there: ROS production, survival, and tail fin swelling. And tail fin swelling. So you can see that any compound that reduced ROS levels to 33% or lower. So we set the threshold at 33%. 33% or lower would be considered a hit. If the same compound can improve survival and reduce tail fin swelling, it would be considered a hit in this zebrafish drug screen. So one such hit that we got was drug U. And I'm going to show you how it rescues all those phenotypes that I was talking about. So tail fin swelling, again, LPS treated larvae show tail fin swelling compared to the controls. You can see that in the presence of the drug, this tail fin swelling is rescued. To visualize this better, we use transgenic zebrafish lines with green blood vessels and injected quantum dots this time. So quantum dots are microangiography contrast agents, and they are the size of your RBC. So they're bigger than phytidextrin. And you can see that even those quantum dots will leak out of the blood vessels. It shows how big the blood vessel leaks are. And these quantum dots move all the way to the tail fin and accumulate there. And this is what you see here. So in the presence of the drug, you can see a complete rescue of this phenotype. Then moving to the third readout, ROS production. Again, you can see that the LPS treated fish light up in green. And in the presence of the drug, you can see that this effect is rescued. The drug also improved survival in a dose-dependent manner, as shown in this graph on the left. So now we identified a drug that is able to rescue the phenotypes that we had established in zebrafish sepsis model. So the ROS phenotype, the mortality, as well as the vascular leakage phenotype. Now the question is, how is it acting? So because vascular leakage is such an important component of sepsis, and because it's increased in sepsis, we thought, okay, it must be acting through the cellular junction proteins. So we measured the expression levels of cellular junction proteins, and you can see that LPS, shown in red, reduces cellular junction protein levels, occluding V, cadherin, and SO1. And in the presence of the drug, shown in green, you see a reversal of this effect. Now, coming to cytokines, we show that LPS can increase the expression levels of all the pro-inflammatory cytokines we tested, interleukin-1, TNF-alpha, IL-6. And in the presence of the drug, shown in green, you can see that we get a rescue here as well. So we established that the drug is effective in zebrafish sepsis model. Now we need to move it forward. And this is the translational part of this research. So the next thing we did was we tested it in mice sepsis models. So CLP model for sepsis is a clinically relevant mice model for sepsis. It's called cecal ligation and puncture. So essentially what you do is you take out the cecum, you ligate it, and then you make holes in it. So when you make holes in it, the contents of the cecum will spill out into your body cavity, and this will induce polymicrobial sepsis. So the CLP mice that received the vehicle, shown in red, showed increased protein leakage in their lung. And the CLP mice that received our drug, you can see that there's reduced protein leakage in the lung. When you look at survival, the CLP mice that receive the vehicle show increased mortality over time, while as the CLP mice that receive the drug, shown in green, have an improvement in survival over time. Finally, we tested this compound or this drug in vitro animals, in vitro human cell models. So we tested it in HUVEC cells, and we used a transwell system for this. So this is a 24-well plate, but within each well, you have an insert. And the insert has a membrane at the bottom. So you're going to grow your cells on this membrane, and you grow it so that it is nice and tight and forms a nice monolayer. Then you add your treatments into the top chamber, so LPS, along with phytidextrin. And then you periodically take samples from your bottom chamber and you measure fluorescence in the bottom chamber. So if there are leaks, between the cells, your phytidextrin will slowly move into the bottom chamber. And you can see that's what happens in the LPS treated larvae. Over time, you can see that the phytidextrin leakage increases over time. And in the presence of the drug, shown in green, you see a rescue of this effect. We also measured the transendothelial electrical resistance across these two membranes. And if the resistance is low, it means that there's increased leakage. So you can see that in the LPS treated cells, there's low resistance and increased leakage. And this, again, this effect is rescued when you add our drug. So this concludes the second part of my presentation. And now coming to the last and final part, the use of zebrafish in personalized human medicine. So these two studies were conducted in collaboration with some groups in British Columbia as well as in Switzerland. And broadly speaking, it looked at sialic acid metabolism and genetic diseases. So the schematic diagram is a little bit heavy. So just remember these important points. So sialic acid is an umbrella term that you use for a family of derivatives of neuraminic acid. And N-acetyl-neuraminic acid that I'm pointing to here is the major sialic acid that you see in humans as well as in many other mammals. NANS is the enzyme that is involved in sialic acid biosynthesis, and NPL is the enzyme that is involved in its catabolism. And when sialic acid is broken down, it is broken down into MANAC and glucanac. So these are the only terms you need to be familiar with. The enzyme NANS that's involved in sialic acid biosynthesis, and the enzyme NPL that is involved in sialic acid catabolism, and the products of catabolism, MANAC and glucanac. So our collaborators did some whole exome sequencing studies, and they identified that in 9 patients that they saw, they all had mutations in the NANS gene. And all these patients showed some common defects or deficits as well. They all had infantile onset severe developmental delay, skeletal dysplasia characterized by abnormally shaped bones, especially in the head and the jaw. And so we thought, can we model this mutation in the zebrafish? So in all these 9 patients, NANS gene is affected and it's knocked out or knocked down. So we thought, let's model NANS gene knockdown in the zebrafish. We used NANS morpholinos, and we used a staining technique called Alcian blue staining to stain for cartilage and bone mineralization. We saw that the NANS morphins, seen here, display a small head, pericardial edema, and abnormal cartilage and skeleton formation when you compare it to the controls, especially in bones that make the jaw. So these are all the bones that make the jaw. And a striking finding was the hypoplastic or absent Meckel's cartilage. So Meckel's cartilage is this bone that I'm pointing to right in the front. And you can see that the controls have a complete Meckel's cartilage, while in the morphins, it's completely absent. So the next step was, so we know that NANS gene is an enzyme involved in sialic acid biosynthesis. So we want to know, can we add exogenous sialic acid into the zebrafish water and rescue this phenotype? So I want you to look at the 3 phenotypes at the bottom. Phenotype 1 is, or P1, is the healthy phenotype. It's the phenotype that has the complete Meckel's cartilage. Phenotype 3 is the phenotype that you see in most of the morphins, and it is the worst phenotype that you can see. It has a complete, the Meckel's cartilage is completely lacking in the morphins. Phenotype 2 is kind of in between. You see a partial Meckel's cartilage in these fish. So now coming to the graph, you'll see that in all the controls, non-injected control, as well as this fish that were injected with control morpholino, you see that the majority of fish, 99% of the fish, were healthy. They had the P1 phenotype. They had a complete Meckel's cartilage. Now in the morphins, you'll see that most of them had the P3 phenotype, shown in green, followed by the P2 phenotype, shown in orange, and then very few of them had the P1 phenotype. Now look what happens when sialic acid is added into the water. You'll see that it's a partial rescue. A lot of them become healthy. So a lot of them develop the full Meckel's cartilage, and there are some that are still unhealthy because, like I said, it's a partial rescue. So this particular study shed light on potential therapeutic strategies that we can use to counteract NANS deficiency in these patients. So provide a more personalized type of medicine for these patients. So now we have established that sialic acid biosynthesis is very important. How about its catabolism? So again, our collaborators did the whole exome sequencing studies, and they identified biallelic NPL variants in 2 siblings, 2 Filipino siblings. That's what's shown here. So at position 133 of the NPL gene, adenine was replaced by guanine. It's a missense mutation, so your amino acid will be completely switched up. So asparagine, which is conserved across species at this position, gets replaced by aspartic acid. Then at position 187, you see that cytosine is replaced by thymine. And again, it's a missense mutation, so the amino acid also changes. Arginine, which is completely conserved across species at this region, gets replaced by a cysteine. And both these siblings that had this phenotype, they exhibited some symptoms as well. Cyalluria, meaning increased sialic acid in the urine, exercise intolerance, and muscle wasting, along with cardiac symptoms. And both of them had these mutations in the NPL gene. And these two can be mapped to exon 2 and exon 3 of the NPL gene. So our first challenge was to see if NPL is expressed in zebrafish, because like I said, only 84% of disease-causing genes have a zebrafish counterpart. So we want to know first if NPL is even there in the zebrafish. So for that, we did in situ hybridization. And to design a probe for an in situ experiment, you need to design an antisense probe that is complementary to your mRNA of interest. So in our case, NPL. So you can see that, and expression is shown in blue. So you can see that from the first 1 cell stage, itself, you can see high level of NPL expression. Then when you move on, you can see a lot of it, the expression is in the muscles. So you can see NPL is highly expressed in the muscle somites. Now, whenever you see a high level of expression of any gene in any tissue, it means that gene is very important for some function in that tissue. So that this was a good sign. We know that NPL is expressed in zebrafish, and it's expressed in the right spot that we were looking for, in the muscles. So the next step was knocking down NPL. And again, we used morpholinos to knock down this gene. And we saw that this resulted in severe skeletal myopathy and cardiac edema. So these are the muscle fibers, and we used phalloidin staining for this. You can see that in the controls, it's a nice arrangement, a symmetrical arrangement of fibers, whereas it is disrupted in the morphins. When you look at the heart, you have the ventricle here and the atrium here in the controls, and it's looped. So zebrafish heart is undergoes two processes called looping and jogging during embryonic development. And both these processes are okay in the controls, but when you look at the morphins, you'll see that it has affected this heart looping. Next, what we tried to do is we used monosaccharide replacement therapy. So we added the catabolic products of sialic acid breakdown, so MANAC and glucanac, into the water. And you can see that MANAC was able to partially rescue the muscle phenotype as well as the cardiac looping defect that we see in the morphins. Glucanac, on the other hand, was only able to partially rescue the muscle phenotype. And if you add sialic acid directly, you don't see any effect. Actually, it makes things worse and that's what's expected, because these patients suffer from an excess of sialic acid in their body. So if you add more sialic acid, it's only going to make things worse. So from this presentation, I hope that I was able to convince you that zebrafish are excellent model organisms for medical research. So we first talked about how zebrafish disease models can be generated either through genetic manipulation or by chemical induction. And once the model is generated, you can use it in high throughput drug screens, phenotypic screens, to identify hits and leads that can be further optimized for preclinical efficacy. It goes through clinical trials and it can reach the patient. For the personalized medicine aspect of zebrafish research, you start at the patient. Human genetics will identify mutations in these patients that are responsible for a particular disease or illness. And then we model that mutation in the zebrafish through genome editing. Again, we create zebrafish disease models for that disease, run it through phenotypic screens, identify hits, preclinical efficacy, test for preclinical efficacy, go through clinical trials, and it goes back into the patient. So in all these ways, we can effectively move a zebrafish from its original aquarium tank all the way to the patient bedside. And in this way, we can effectively advance drug discovery. With this, I conclude. I would like to thank you all for your patient listening, and I would be open to answering questions at this point. Thank you.
Ma'am, a million sincere thanks to you for the sea of wisdom and knowledge you showered us through your amazing lecture. You're too excellent, ma'am. Thank you so much. I'm sure that everyone enjoyed this amazing presentation. So now, ma'am will answer some of your questions. Dear participants, please be sure to type your questions in the chat box. Can we go ahead, ma'am?
Yes, you can go ahead.
Okay, the first question is that in atorvastatin-induced ICH model, is it the fluorescent model, or what method is used to detect the hemorrhage?
So okay, just 1 second, please. So my son has actually come into the room. Okay, so in the atorvastatin model, we use those double transgenic lines again. So we use the fish having the green blood vessels and the red RBCs. So when we put them in atorvastatin through the embryo water, you can see that the RBCs will leak out of the blood vessels. So the blood vessels break or they leak out, and it's the RBCs that you see accumulated in the brain region.
Okay, ma'am. Okay. The next question is any zebrafish model available for screening of anti-Alzheimer's agent?
Yes,
Screening
there
of
are. Yeah, so there are Alzheimer's models. So like I said, there are different techniques in which you can make this. You can do genome-wide association studies and find some genes that are relevant to Alzheimer's, knock it out, and then check zebrafish after the knockdown. So for these neurological diseases, the problem is what are the readouts that you're going to use? So sometimes, like in Alzheimer's, depression, all those kind of diseases, what we do is we check for zebrafish behavior. There are apparatus and techniques that we can use to check for zebrafish behavior. So sometimes when a fish is depressed, it will just swim in the bottom layer of the tank. It will not go all over the tank and swim, because usually zebrafish are very curious. They move all over the tank and they swim around the tank. If you add food, they'll go and get it from the top. But if they're depressed, if you knock down some of these genes, they might get depressed. And that a way to measure depression is they won't move around that much. They just kind of stick around at the bottom of the tank. So this is something that we do in our lab as well. We do check for behavioral phenotypes.
Okay, ma'am. The other one is whether the zebrafish is a good model for cancer studies. How about your opinion?
Yes, it's an excellent model for cancer studies. Like I said, you can use the CASP. I don't know if you remember, I showed you a transparent zebrafish line, right? In one of the slides, it's called Casper line. You can even visualize tumor metastasis through this line because it's transparent. So where the tumor starts, where it's moving, and everything you can visualize in the zebrafish. So it's an excellent model for cancer studies.
Okay, ma'am. The other one is how to increase zebrafish survival rate after spinal cord injury.
Zebrafish survival rate? I didn't quite understand the question.
Maybe the question. Okay, now let's move on to the other one. Okay, ma'am. Maybe what the person mean, I don't understand.
Maybe it's their meaning can be model zebrafish spinal injury. Maybe that's what they're meaning in zebrafish. If that is the question, yes, you can model spinal injury in zebrafish. We actually have models in our lab where we are mimicking traumatic brain injury and spinal cord injury in the zebrafish.
Okay, ma'am. There's a person asking, is there any other fishes which can be used in research purposes like zebra banyo?
I think Medaka is used a bit in research, but not as much as the zebrafish. Zebrafish have gained a lot of widespread acceptance as well by the research. So initially people were a little bit skeptical about zebrafish, but when we actually show that these drugs can be moved to the bedside, the drugs that we discover through zebrafish drug screens when we actually show that it can reach the patient and it can move directly to the patient, that's when the research community started accepting zebrafish as a model for research. So I don't think there's any other fish model that is as accepted as the zebrafish model.
Okay, ma'am. Can you explain once again on loping and jogging?
Yes. Yes. So during zebrafish heart development, the heart tube, it moves to the left first, and that's the process of jogging. That's when the ventricle is formed. And then to compensate that, the atrium shifts to the right, and that's the process of looping. That's why the heart looks looped. It's two-chambered, and so these are two important processes in zebra: jogging and looping.
Okay, ma'am. And what are the physical factors that affect the developmental stages of zebrafish?
The physical so the water quality, the pH of the water, everything has to be maintained. So we have tons and tons of zebrafish in our lab. And often when the pH changes, we get an alarm signal. So any of this can affect their reproduction, like how well they lay eggs and the quality of the eggs as well. Also, if you're not using zebrafish for breeding for a while, they say that you should, so usually we keep zebrafish males in one tank and females in one tank. And then right before breeding, we put them together the night before breeding, and then they breed. But if you're not using them, they say that you should put the males and the females together so that they can get rid of their old eggs and all that. Because if they don't, what happens is that the zebrafish females stop laying eggs because they have this old supply of eggs in their body and they will not produce any more eggs. So that's another physical thing that we do, that we separate them out into 2 different tanks. But if you're not using them for long, you have to put them together so that they can continue their natural process of breeding.
Okay, ma'am. There is a question related to the recent times, that is it applicable for COVID, drug against COVID?
Actually, I don't know if you know, but many of the symptoms of sepsis is similar to what you see in COVID. You see vascular instability, you see vascular leakage, you see leakage in the lungs, especially. So it is applicable to COVID, that you can model. There are many viral diseases that you can model in the zebrafish. So you can model COVID in the zebrafish. And of course, it's the advantage is that you can use it for high throughput screens. You can screen for hundreds of drug compounds against COVID.
Okay, ma'am. The other question is, how easy or difficult is to do CRISPR case-mediated knockout in zebrafish?
So when you compare it to morpholino is quick and easy, right? All you need to do is you need to synthesize the morpholino, you inject it, right away you get the results. CRISPR, on the other hand, it takes more time because you need to wait through generations. You have to make sure that the gene that you are knocking out is indeed knocked out. You have to go through F1 generation, F2 generation. Yeah, like you have to wait a bit to get the results. And knock-ins are possible, but knock-ins are more difficult to do than knockouts. That's what we see when we actually do it.
Okay, ma'am. Now I think we have covered almost our questions in the chat box. Is the other person want to ask our dignitaries research?
Question, it is posted here by Harish Kumar. After treatment with new drug and their recovery, any mortality or any other behavioral changes in the Anju ma'am, you are mute.
Okay, so for after treating with, sorry, I didn't get your question.
After treatment with new drug and their recovery, did you notice any mortality or any behavioral changes?
So sometimes when we start the drug screen, sometimes the drug, the concentration we use is too toxic and the zebrafish will die. So we usually do 3 or 4 different concentrations of the drug when we are doing the drug screen. So it is possible that some drugs are toxic in the zebrafish and that they can die. So yeah, we do see some mortality.
Okay, another question by the same person. What is your current research going on in zebrafishes?
So I am involved in this. So my main project is the zebrafish sepsis model project. So I only talked about one of the drugs that we found in our screen. There are a few other ones that we also identified. So there's more research going in that direction. And I'm also involved in many collaborative projects that they do in the department as well.
How was drug or quantum dots administered to zebrafishes?
Okay, that's a good question. I forgot to mention that. So quantum dots or phytidextrin, anything that you're injecting, you inject into the PCV, the posterior cardinal vein. So I don't know, I showed you in the video the major blood vessels in fish. So you can inject it into the PCV. There's also a common cardinal vein near the heart that you can inject it to. So all these compounds, quantum dots, phytidextrin, anything you want to inject, after the larvae is hatched, you will inject directly into these blood vessels.
What are the steps followed in creating a zebrafish model? Can you explain with example to human disease?
well, I think that was what I was explaining, right? The steps in creating a model. So the first thing is, of course, identifying, like if it's a mutation that causes a disease, you identify what the mutation is, in which gene is affected, then you model that mutation in the zebrafish. And for that, you can use either CRISPR-Cas9 or you can use morpholinos. And then once you knock down or knock out a gene, you look for what the phenotypes that the fish shows and you characterize it. And then later on, you can use it in drug screens.
Okay. How to do inflammation studies in zebrafish?
So sepsis, for example, is an inflammatory disease, right? It's a state of dysregulated immune response. So, and all these cells like neutrophils, macrophages, everything is there in zebrafish. And you even have transgenic lines for zebrafish. So you have transgenic zebrafish lines where the neutrophils appear green, the macrophages appear red. So you can use all these models to study inflammation.
Okay. What's your thought on ethical issues associated with using live organism for research?
If it's necessary, we need to use organisms in research, right? And with the zebrafish, I personally don't feel as bad when I'm using zebrafish because they lay hundreds and hundreds of eggs at a time. So previously for my PhD, I was working with rainbow trout, and they are huge fish. So when I had to sacrifice a rainbow trout, I was very traumatized. The night before, I couldn't sleep when I had to sacrifice rainbow trouts because I feel that they're big and I don't know, I feel a bit traumatized. But in the case of zebrafish, because they lay hundreds and hundreds of eggs at a time, they're really small, I don't feel that feeling. And I feel that if it's a way to advance research, we need to use model organisms in research. But of course, if you can use zebrafish in place of mice, always go for zebrafish, because the more higher the organism in the status of the organism, the more problems you get from committees, the animal safety committees and all that. But zebrafish, there's not much issues with using zebrafish in research.
Yes, that's right. Can we induce inflammation in gut by LPS in adult zebrafish?
Yeah, so I put LPS in the water, right? But you can always do LPS injections into the fish as well and induce inflammation in them that way as well. This is the most easy method. Putting drugs and treatment in the water is the most easy method, and zebrafish are very permeable. So they can get these treatments into their body either by swallowing or through their skin. Their skin is very permeable. There are also agents that you can add into the water to make it more permeable. For example, there's DMSO that we can use in the zebrafish water. And as long as the DMSO concentration is less than 0.01%, it is not toxic to the zebrafish. And this will further permeabilize the zebrafish skin so that you can get the small molecules to enter into the body. But like you said, yes, you can do direct injection of LPS into the zebrafish as well.
Okay, more questions are there. Zebrafish lack the complexity of advanced cognitive issues. How is it then possible for a successive study in neurodegeneration?
So that is one problem with zebrafish, that you say that how can you say that a zebrafish is depressed, or how can you say that a zebrafish has traumatic brain injury or something like that? So that is a problem that we always face, or a criticism that we always face. But we measure these things, like I said, for depression. A heat map of the area in the tank that the zebrafish swims in. So we compare between controls and the more mutated ones, the heat map of the area in the tank that they swim in. So controls, it's all over the place. The ones that we knock down the gene for like neurological diseases, they're very depressed. They sit at the bottom somewhere. And so some stuff like that, we just have to change the readout that you look at. And use ones that are more accommodating of the zebrafish lifestyle and use those kind of readouts in our studies.
Okay. Another question, can you explain TNBS modeling in wild type fish? It's regarding wild type fish. TNBS modeling.
So wild type fish, we have lots of wild type strains. There's the TU strain, there's the Leo strain and all that. But obviously we are using transgenic lines for our studies. So those are the lines that are more effective in our studies when we knock out the genes and create transgenic lines for all the different processes like cardiovascular functions. You can create transgenic lines, immune relevant genes like cytokines, all of those you can create transgenic lines.
Okay. Another question on coming. One sec. Another question is one sec. I saw a genuine question. Wait. It's from YouTube only. So many questions are there taking it from YouTube. So based on your studies on human drugs in zebrafish, how efficiently is it reflected in higher models like mice?
So like I said, because we are using an in vivo, so the advantage of using zebrafish is that in the drug discovery pipeline, even from the start, you are using an in vivo model. So I feel that any drug you identify through an in vivo model will always be more effective when it comes to the clinic. When you actually go into clinical trials, it's always going to be more effective rather than just modeling it in a cell system. So for like neurological diseases, by using neuronal cells. Instead of that, if you use the full organism, you get a bigger picture of the disease. And at the same time, you get you can model the disease correctly, like all the processes of the disease correctly, and you can get better targets, drug targets as well. Drug hits as well, sorry.
Okay. Which mammalian cell lines are used in this experiment-based research? CHO or HeLa, which one?
No, we used HUVEC cells, so human umbilical vein cells. So we have also used HPMECs, human pulmonary microvascular endothelial cells as well. But I haven't used HeLa cells in this research. What I showed is just HUVEC cells and HPMECs.
Another question by the same person who asked about LPS. If we are adding LPS in water, can we be able to say particular inflammation in gut alone?
No. If you add LPS in water, what you get is a systemic inflammation because it's entering through the whole body, right? All the skin and everything. If you want to look at gut alone, you have to inject directly into the gut itself. You cannot use LPS in water. What I wanted was a systemic inflammation, whole body inflammation. I wasn't targeting any particular organ.
The study states that zebrafish have regenerative capacity in their heart. Can we identify their genes and implement in human gene therapy?
So this is a good question because it's very true. Zebrafish are very well known for their regenerative capacities. If you cut out their heart, in a week's time, they will regenerate 40% of it back. So we know this happens, but we don't know how this happens. That's the problem, and we're still studying it. When we check these zebrafish that regenerate their heart and other tissues back, we know that the cells are coming back to a de-differentiated state. So you know that cardiac myocytes are all differentiated. But when they're in the process of regeneration, they all go back to a de-differentiated state. But we still don't know what are the cues that lead to that. And that it is still an active area of research. Of course, if we can find something in that regard, it will be extremely useful in humans.
Okay. Another question, how can you experiment skin inflammation studies in zebrafish? Or can you use other model?
There are melanoma models, like melanoma cancer models. Then there are also, so zebrafish has 3 different skin pigments, like black, reflective, and yellow. And you can try to mutate these skin pigments. And one mutant strain, like I said, the Casper strain is one that lacks all of these 3 pigments except one. So stuff like you can use zebrafish for anything to study. If you know that what causes a particular disease, you can. First thing is you have to make sure that zebrafish have that gene, or they have the same gene. If it has it, you can definitely mutate it and study anything. You can study skin diseases, inflammation, anything.
If we are adding this photo, that's all. I think we have addressed almost all the questions. If in case if we have not, we can respond to you through your email. If we have any other queries, also we can address through your email ID. So participants, kindly excuse me if at all couldn't address any of your questions. So we'll address those through email. So that's all. Over to you, Niduna.
Okay. Thank you again, ma'am. Anju, ma'am, thank you so much for answering those questions and for the great presentation. It was a pleasure to have you with us. Okay, ma'am, thank you so much. So now this concludes the webinar. Thank you all for attending. We hope you have learned and enjoyed this presentation very much. Now I invite Dr. Serene Sarah John, ma'am, faculty coordinator, UC College, for the vote of thanks. Ma'am, please.
Okay. A warm and graceful day to all our distinguished guests, participants, and faculty members. The Department of Biosciences has been conducting many webinars during the past few months, and we have conducted a Global Biotech Week from September 28 to October 4th with 2 national webinars, competitions like logo designing, poster designing, article writing, quiz, etc. After that, we conducted a 2-day webinar on machine learning for biology, especially for the students of bioinformatics. And on October 15th, we organized another webinar on the hottest topic, climate change research and resilience. So right now, I am very much delighted that we could conduct today's webinar jointly with UC College Alumni Association of North America. And same time next week, on October 31st, we have one more webinar in association with UCCAANA. The webinar is on molecular dynamic simulations in computer-aided drug discovery by Meenakshi Sundaram Balasubramaniam from University of Arkansas. And 2 more webinars are on novel topics are on the pipeline. And with this brief information, let me begin about our activities. Let me begin my duty of saying vote of thanks. So I must compliment our speaker, Dr. Anju Mary Philip, for a wonderful session with amazing videos. Like it was lucid, simplified, and yet another impactful session. And I know the depth you have in this field. It was so excellent and a cohesive presentation. We had a great learning opportunity. I think many of the participants have been encouraged by your presentation and everything. And on behalf of the organizers, Department of Biosciences, and the entire fraternity, I extend my hearty thanks to Anju ma'am. Our beloved manager, Reverend Father Thomas John, a great visionary, is always among us, and we thank him for being the catalyst that stimulated us to do the best part as part of the centenary celebrations of the college. With deep sense of appreciation, we thank you, sir. And in spite of the absence of our beloved principal, David Sachs Matthew, sir, I thank him for being with us always as a pillar of strength. Then I am grateful to all the UCCAANA members, especially Dr. Aipurgi, sir, for expressing the willingness to associate with us and for providing the right speaker for this webinar. Looking forward to have more associations with with the UCCAANA members. Okay, then An event like this could not have happened in an overnight. The wheels start rolling weeks ago. It requires the planning and bird's eye for details. We are fortunate enough to be backed by Dr. Susan Epen, our adjunct faculty, of course, a very proactive and dedicated person, and a dynamic person also. Thank you, ma'am, for your excellent support. My sincere gratitude goes to our head of the department, Shyam Mohan sir, for the perfect support and guidance extended. On behalf of the entire fraternity, I extend special thanks to Shyam sir for his constant encouragement. Now I convey my gratitude to our director, Dr. Alex sir, the faculty members of the college, retired faculty members of the department, my colleagues, and all the students of the department. Also, I must thank the students who took pains to throw such a wonderful event for us. Special thanks goes to Ashwin, Niduna, Akhila, Anita, Maria, and all who coordinated the entire event. Last but not least, I thank all the participants for showing their genuine interest to attend this program. Thank you once again. Please do join us for the next webinar to be held on 31st of October, 6:30 p.m. That is Indian Standard Time. By then, see you, take care, and stay safe. That's all. Thank you. Thank you all. Thank you.
Thank you, ma'am.
Thank you, ma'am.
Bye-bye.
Bye.
Thank you, Anju.
Thank you, madam. Thank you.